Okay, this particular EdPuzzle is going to be about viruses, what they are, how they hijack our cells to copy themselves and all the kinds of damage that they do, and then a little bit about what we can do about them in terms of either drug treatments or how our cells can naturally resist them invading. First of all, what is a virus? A virus is not a cell. A cell we have defined as the basic unit of life. Viruses are much smaller in general than cells, much smaller. And a virus needs a cell in order to be able to replicate. So the definition of a virus is essentially an infectious particle that generally consists of some genetic material. This could be DNA, this could be RNA, that's surrounded by a protein coat that we call a capsid. In some cases, the capsid is itself surrounded by a double layer of cell membrane. The cell membrane is membrane that the virus stole from its host cell. And is using it as sort of like a Trojan horse or a cloak of invisibility to be able to get into the next host cell, and we'll talk about that more here in just a minute. So a virus, by definition, cannot carry out it doesn't carry out really any metabolic activity, and it cannot copy itself, and it cannot really live outside the host cell. When we talk about a virus, people talk about live virus. It's not alive because it's not a cell, right? It's what we refer to as basically an obligate cellular parasite. So it's not a cell, but for something that is not technically alive, they sure do cause a lot of problems. So a virus can be infectious and stay on surfaces and body fluids and things like that. To where it has the ability to enter other cells and infect them, essentially. And so that's that infectious period, or that period that we say a virus is live, is basically how long can it infect you if it's outside of a cell. But a virus just sitting on a tabletop or something like that is completely inert. I mean, it can't hurt you. It only can hurt you if it gets inside your cells, and that is the problem, right? We want to try and keep these little buggers out of our cells because what they do once they invade our cells, and which we're going to see here in just a second, is essentially hijack all of our DNA replication machinery. They hijack our ribosomes to make viral proteins instead of our cellular proteins or bacterial cellular proteins. And essentially they take over the cell and force the cell, it's sort of like mind control, force the cell to replicate the virus instead of doing cellular activities, right? So basically the virus comes in and takes over and says, hey, you're just going to make more copies of my genetic material. You're going to make my protein capsid, etc. And I'm going to self-assemble, and then I'm going to leave, and I'm going to try and get into more cells and more cells and more cells. So which is why we refer to them as an infectious particle. It's basically a cellular parasite. And of course, as we've seen, right, they're really tiny, and most are too small to see under a light microscope. Both of these images are electron microscope images. So here we have a picture of a couple of different types of viruses. Viruses really vary in terms of their appearance, but their general makeup, as we were saying before, is essentially genetic material. Again, this can be linear, like our chromosomes. They can be circular, a loop. They can be made of DNA. They can be made of RNA. They can be, again, quite often you have the genetic material wrapped in that protein coat that we call the capsid, right? The capsid then can sometimes it's just the protein coat and the genetic material, like the virus on the right here, is very typical of what a bacteriophage looks like. A bacteriophage is a virus that specifically infects bacteria, as the name implies, right? So a bacteriophage cannot infect a eukaryotic cell, right? So we are completely safe, for example, from bacteriophages because they don't have the mechanisms to infect our cells. They only infect bacterial cells and quite often infect very specific kinds of bacterial cells. General mechanism for for viruses to infect a host is injecting their genetic material into the host, so the capsid stays outside, and what enters the cell is just the DNA or the RNA and any other enzymes or molecules that are contained inside the capsid. Viral genomes tend to be much smaller than bacterial genomes, which are often several hundred genes or to a few thousand genes. Eukaryotic genomes are much, much larger. We have, for example, humans, we have, hundreds of thousands of genes. So viruses in general are much, much simpler in terms of their DNA or RNA structure. The virus on the left in this picture is an example of a virus that is going to enter the cell by means of what's called a viral envelope. That's that circular part with all the little sort of look like thumbtacks sticking up out of the surface. Those are called glycoproteins, and basically they help the virus to essentially decode the receptors on the surface of the host cell and fool the host cell into allowing the membrane coat to merge with the host membrane. And of course, once you do that, right, then you can dump the contents, which in this case is the capsid, into the host cell. The capsid dissolves, and then you have the virus, basically naked virus. It's just its genetic material and any other molecules that it's brought with it. And that way it's gotten into the cell without injecting its material. Instead, it's merged its membrane coat that, as I mentioned, it stole from its previous host. So they don't make their own membranes, right? Viruses don't make almost anything themselves. They steal them or get cells to do their bidding. That's essentially what they're all about. So he's managed to merge that membrane with the host membrane, and that's how it's getting inside. So you're either injecting it and leaving the capsid outside, or you have an envelope that merges with the host membrane, and then the capsid dissolves and reveals the virus inside. Okay, so as I mentioned, a virus, right, cannot replicate outside of the host cell. It's completely helpless in that regard. Now, again, not to say you shouldn't be respecting viruses, right? But they can only do damage if they can get inside their host cell, right? Whether their host is a bacteria or whether their host is a eukaryote is, of course, going to be different for each type of virus. So a particular virus generally infects a limited number of host species, right? And these could be types of bacteria, or it could be types of eukaryotic cell. And the reason for that is we have an arms race going on, right? Our cells are constantly, and bacterial cells are constantly mixing and matching our surface receptors to try and keep the viruses out, try and keep them from penetrating our host defenses. In particular, this is true of bacteria. They sort of really scramble their receptor proteins quite a bit, trying to resist their bacteriophage parasites. So the surface proteins are going to identify receptor molecules. The surface proteins of the virus identify receptor molecules on the host cell surface. So they're going to be fairly specific. So for example, bacteriophages will infect only 1 or 2 at most types of bacteria. And they're fairly specific in their function because, again, those protein receptors have to match. And they're constantly changing, so the virus has to constantly change as well to try and keep up. In eukaryotes, for example, West Nile is carried by mosquitoes. It does not actually infect the mosquito. The mosquito is just the vector. But it infects basically birds and humans, basically people. It's been known to infect some other mammals and predominantly some other bird species. So the rhinovirus, for example, pretty much just infects our respiratory tract. So it's specific to people. And quite often other species have their own version of, those kinds of viruses. So again, our virus has basically 2 main ways of getting inside the host cell. One is by injecting its genetic material into the host, usually through those surface proteins that are going to recognize the virus and allow it to inject its DNA, or through the use of the viral envelope, right? So quite often the code for the surface receptor proteins is contained in the viral DNA or the viral RNA that infects the host cell. The host doesn't have instructions on how to make those surface proteins. But the membrane itself, which is basically just a lipid bilayer, right? That the virus is getting from its host if it's using a viral envelope to infect its host. So once it's inside, right, it's going to hijack the cell's machinery and say, hey, you're not going to make your stuff anymore. You're going to copy my genetic material, and you're going to make my capsid protein, and you're going to make the receptors for the viral envelope. And then, of course, it's going to steal the membrane and wrap it around the capsid, which the capsid is in turn wrapped around the genetic material. And now you have an infectious viral particle that can leave the host cell, again, by merging with the membrane and then leaving as this little kind of encapsulated blob. And it's leaving with those receptors on the surface. And those receptors then allow them to fool the next host into allowing, right, the virus to merge membranes and then dump its contents inside, which is the capsid wrapped around the genetic material, along with any other, enzymes or whatever, the viral enzymes that are coming in, and now you've infected the next host. So again, these types of viruses that use an envelope are generally going to be ones that infect eukaryotic cells, because they often will use either the cell membrane. Sometimes they'll use organelle membrane, but for whatever reason, they tend to infect eukaryotic cells rather than bacterial cells. Bacterial cells are often the victim of having that genetic material from the virus injected into them, and the capsid stays outside. So for viruses that inject their DNA into their host, which of course is going to be generally bacteriophages and some eukaryote viruses, they either inject their genetic material into the host or they trick the host, again, by matching those surface recognition proteins. They trick the host into forming a membrane vesicle, basically a little membrane bubble around the virus and bringing the virus into the cell, capsid and all. No membrane envelope though. So they enter by endocytosis, and once that happens, right, once you have the virus inside the cell, the viral genome then commandeers the host replication machinery, right? So now the host cell is copying viral DNA, right, or viral RNA instead of copying its own genetic material, and it starts making viral proteins, right, because you can then make messenger RNA and send that to the ribosome, and then you start making those viral proteins. Typically what happens in the case of viruses that inject their DNA or RNA into the host cell is that they replicate their genome a whole bunch of times, maybe thousands of times, and that the genetic material and the protein capsid spontaneously self-assemble to form a new virus. That's a new infectious virus. And then basically this keeps going until the cell is so chock full of infectious virus particles that the cell explodes, right? So the cell lyses, and this allows the now newly infectious virus to leave the host cell, which is now dead. And that's something we refer to as the lytic cycle, right? Because the cell is lysed in the process. Viruses that perform this particular type of process are the most destructive, as you can imagine. The cells are destroyed in the process of viral replication. So you can imagine that there's tremendous selective pressure on bacteria and on eukaryotes to resist invasion by these types of viruses, right, that are going to destroy the host cell. Part of that is done through this changing of the surface recognition proteins such that the host cell doesn't recognize the virus as somebody who it should allow in, so it doesn't form an endocytosis bubble or membrane bubble and bring it into the cell, or it doesn't allow the virus to inject its genetic material into the host because, again, doesn't recognize those surface proteins. Other ways that cells can resist invasion by these type of we refer to as virulent phages if it's infecting a bacterial cell, or a virulent virus if it's a eukaryotic cell. Another mechanism of defense is essentially using enzymes to identify and destroy the genetic material. So we have, and in eukaryote cells we have this as well. We just don't generally have as many of them floating around. Bacteria have a lot of virus parasites that have a lot of phages to have to deal with, and so they tend to have a lot of these restriction enzymes. Restriction enzymes recognize specific DNA sequences and will cut the DNA, or in some cases RNA, at specific locations. Essentially, they're like using scissors on the phage DNA and will chop it up into pieces. And of course, if it's in pieces, then it's much harder to copy it. It's not going to take over the cell, and basically the bacteria has succeeded in defending itself. So either you keep the virus from getting in the first place, or you try and destroy the DNA of the virus once it's come inside. Now, our cells have similar types of enzymes. We actually have hydrolytic enzymes that again are breaking down RNA in particular inside of our cells, and we're going to talk about this later on when we talk about transcription and translation, because when you're making protein, generally what happens is you make a copy of the DNA of the gene that you want to express, the protein that you want to make, basically the instructions. You make a copy in RNA, and that's what goes to the ribosome. But the ribosome can read that RNA copy, the instructions on how to make the protein. It can read that over and over and over again and keep making the same protein over and over and over again. Sometimes that's what our cells want. They actually want to make lots of copies of a protein, especially if it's something we really, really need. But again, we have enzymes that are essentially degrading that messenger RNA molecule by breaking it down on one end and sort of chewing their way up the RNA molecule. And part of the function of those enzymes is to identify, in particular, RNA that is not ours and break it down. So again, trying to protect us from viruses that are infecting our cells. So a virus that uses only the lytic cycle, that's only way it replicates itself is by invading the host cell and making so many copies of itself that the cell dies as a result of the virus's leaving. We refer to as a virulent phage if it's infecting bacteria, right? Or just a virulent virus if it's infecting a eukaryotic cell. So an example of a virulent virus would be something like Ebola, where it injects its DNA into the host cell. Actually, I think it might do it by endocytosis. Either way, the DNA and the capsid get inside, and then it takes over the cellular machinery, copies itself so many times that the cell bursts. And unfortunately, it usually does this in a fairly short turnaround time, which is why, unfortunately, that Ebola is so deadly. So the lytic cycle is one way that a virus can infect and copy itself and then destroy the host cell in the process of leaving. But not all viruses use that particular cycle, right? So some are what we refer to as temperate phages if they infect a bacteria, or a temperate virus if they infect a eukaryotic cell. And these types of viruses use both the lytic and the lysogenic cycles, and they alternate between the two. So the lytic cycle we already know about, right? This is the one on the left. So I get my DNA in there, I hijack the replication machinery, I trick the host cell into making more of me, both copying my genetic material and making the protein capsid. I self-assemble, and then I lyse the cell as I leave. Some viruses do this for a while and then will switch to what's called the lysogenic cycle. And as part of the lysogenic cycle, yes, they inject their genetic material into the host cell, but instead of hijacking the copying mechanisms, they insert their genetic material into the host cell's DNA. So in the picture we have here, we're looking at a bacteria. Bacteria have circular genomes, right? Unlike eukaryotic cells that have linear chromosomes. The chromosomes of bacteria are circular, and you can see the little loop of phage DNA that has inserted itself into the DNA of the host cell. And the idea behind this, of course, is that every time the host cell copies itself, it is copying the virus, right? And so essentially the virus is laying somewhat dormant inside the genetic material of the host, and this can continue for generations and generations and generations. This can continue for a really long time. And then typically what happens is there's some sort of an environmental change that triggers switching back to the lytic cycle for that particular virus. And again, it can be a very long time that it stays dormant in the host genetic material, or it can be a short time. It's typically a longer period of time. And so we refer to viruses that perform, again, like I said, temperate phages or temperate viruses. We also can refer to them as a prophage or a provirus. So that prophage or a provirus is basically a virus that inserts its genome into the host genome and then lets the host copy the virus every time it copies itself, essentially. And that's the lysogenic cycle. So the lysogenic cycle is being inside the DNA, getting copied that way. So the virus is still getting replicated. It's getting what it wants. It's just not killing the host cell in the process, which of course is what happens as a result of the lytic cycle. So at some point for your temperate phages or your temperate viruses, right, that are using both the lytic and the lysogenic cycle, you're going to use the lysogenic cycle for quite a while. At some point, though, you have an environmental event and it varies by virus in terms of what triggers it. But it's going to switch to the lytic cycle. To do that, in bacteria, the prophage will leave the host genome. Essentially, will extricate itself from the host genome and go back to the cycle of, hey, host, you copy me, so many times that I cause you to explode. Now I'm an infectious virus particle, and I can go to the next host and either trick it into bringing me inside through endocytosis, or allowing me to inject my genetic material into the next host cell, and that keeps the cycle going, and so on and so forth. Prophages, as I mentioned, are sort of dormant inside during the lysogenic cycle. They're somewhat dormant inside the genetic material of the host. However, this doesn't mean that the host can't read and make the genetic products that are coded for by the virus, right? So you basically have this extra code in your genome, and especially if you're a bacteria basically express every gene that they have, pretty much, unlike eukaryotes, which we have whole mechanisms for turning genes off to where they are never expressed in a particular cell. Bacteria don't have that mechanism really, and so they are going to, if they have the genetic code, they're going to make the protein product at some point. And of course, this can be bad news for the host. Basically, expressing viral DNA and viral proteins can change the behavior of the host. So for example, E. coli, right? These are bacteria that live in our digestive system. They help protect us from foodborne illness. They help us digest our food better. They're basically all around happy, happy friends to us in general, except that nasty strain, right? 0157H7 is the strain that causes severe food poisoning and can cause death as a result. And of course, we're prone to getting mad at our E. coli. It's actually not their fault, because what is changing the behavior of the E. coli, it's been infected by a virus, and in particular, it's been infected by a temperate virus that is using the lysogenic cycle. So the virus is hiding its DNA inside the bacterial DNA, and the bacteria doesn't know any better. It reads the instructions, it makes the protein product. Unfortunately, the protein product is a toxin that's going to nastily affect not necessarily the E. coli, but the host of the E. coli, which is us, basically their environment that they live in. This toxin builds up and it causes severe illness. Botulism is another example of a strain of bacteria that was infected with a virus, and it now produces botulinum toxin, which causes paralysis. So this also can occur in eukaryotes, right? So we have temperate viruses as well that are using the lysogenic cycle in addition to the lytic cycle, and that virus can incorporate itself into the host chromosome, which of course is our DNA, and lie dormant for a long period of time until something triggers a new round of virus production. In the case of proviruses, which is what we call viruses that are using the lysogenic cycle and have invaded a eukaryote cell as opposed to a prokaryote cell, proviruses, when the new round of lytic cycle production is triggered, they don't remove themselves from the host DNA. They just copy themselves. And so this means that you may still have some cells that have the viral DNA embedded within them, and others that are going through the lytic cycle that don't and are going to die as a result of that cycle of reproduction. So the herpes viruses are sort of famous for this particular strategy. The red welts or red sores that we get on our faces and other skin locations are the result of the virus going through the lytic cycle. You have tissue that's being damaged as a result of that. At some point, it switches to the lysogenic cycle and is hiding in our DNA, right? So every time our skin cells replicate themselves, and our skin cells replicate a lot because they're constantly being damaged, and we're constantly, they're constantly dying too. So every time our skin cells replicate themselves, they're copying the virus. And again, at some point, something triggers a new round of virus reproduction. Basically, you go back to the lytic cycle again, and you're damaging tissue again. We think it may be linked to stress, but it could also be other types of physiological changes that trigger this. So how do viruses that don't do the endocytosis or inject their DNA method, how do they get into cells, right? We talked about this a little bit earlier when we were talking about the structure of different kinds of viruses. So viruses that use viral envelopes. The envelope, of course, is cell membrane. It's cell membrane that they stole from their previous host, which is kind of creepy if you're thinking about it. I'm going to wear the, the skin of my former host. Yuck. Anyway, but the membrane is then, of course, covered with these glycoproteins. And the glycoproteins essentially help fool the next host cell into allowing the virus to come inside. Basically, the two membranes merge together. And the viral capsid is then dumped inside the host cell, as you can see in this illustration here. So viruses that have viral envelopes are going to come in using a viral envelope. They're also going to leave using a viral envelope. So viruses that use viral envelopes do not use the lytic cycle. The host cell is definitely damaged, but the host cell is not killed in the replication process. So you have, again, recognition proteins on the surface of the host cell, and they're going to dock with receptors on the host cell and essentially trick the host cell into saying, oh, this is something I want, so therefore I'm going to allow it to, again, the membranes merge together, and now the virus ends up inside. The capsid dissolves, and now you have viral genetic material, either DNA or RNA in the case of this particular illustration, that is present in the cell. Now that RNA can then get to work essentially and can act as a template for messenger RNA, right, which is going to go to a ribosome and make that's a ribosome that's either free in the cytoplasm to make the capsid protein, or it can also go to ribosomes that are bound to the endoplasmic reticulum, which is where a lot of protein synthesis happens in our cells anyway. And those ribosomes are going to translate the mRNA, and it's going to make those recognition proteins, those glycoproteins, for the receptors for the surface of the cell. So if you're a DNA virus and you're using a viral envelope and you come in and it's pretty easy, right? Because the host cell is going to be using DNA. So you're going to already have enzymes to copy yourself. You're already going to have enzymes that will read you and make a copy of messenger RNA that would then go to the ribosomes and make those viral proteins that we need. It self-assembles, and again, you have this infectious virus that's going to leave the cell. Now, if you're an RNA virus, though, you have a little bit of a challenge, right? Because our cells are not set up, neither are bacterial cells, to copy RNA molecules the way that we copy DNA to make a working copy to send it to the ribosome, right? So we're not set up for that, and we don't have enzymes to copy RNA to make a copy of that genetic material either, because again, we are using DNA as our genetic material, so we just don't have the enzymes for that. So an RNA virus essentially will need to bring its own replication enzymes. Again, these are enzymes that it got the host cell to build for it using those instructions. But it's going to bring those enzymes are those RNA polymerases. These are essentially the same as DNA polymerase. You're catalyzing that phosphodiester bond between the nucleotides, but in this case, the nucleotides are RNA bases instead of DNA bases. And so it's an enzyme that is shaped to work with RNA instead of DNA. So the virus brings that enzyme with it. Now it can copy its genome, right? It can make a copy of that RNA sequence which of course now I can copy my genome, and then that genome, because it's written in RNA, can function as messenger RNA directly. So you can just make copies of yourself and have some copies will be your genetic material, some copies will go to a ribosome and get translated into the protein. They could also, again, use viral enzymes that they bring with them to make their own messenger RNA from their RNA template, quite possibly. And then they could also take their RNA genome and, again, using enzymes that they brought with them, in particular an enzyme called reverse transcriptase. They can take their RNA genome and convert it to DNA. And this means that now, oh, now I've got DNA, right? Now I can get the cell to copy me, because our cells have DNA polymerases that we use all the time to copy our DNA. So I can get the cell to do that for me. And then I can get the cell to read that DNA and make messenger RNA, right? And you see where I'm going, right? Go to the ribosomes and make the capsid proteins as well as those surface recognition proteins that you're going to need for your viral envelope when you leave. So these are retroviruses. Now, because the virus now is in, well, it's going to copy the DNA to DNA, but also, again, we have enzymes to copy DNA to RNA, so now it has a way of copying itself in the RNA form. So again, you can copy yourself completely. You can do all the translation to make the proteins that you need to infect the next host cell in this method. But while you're in the DNA form, right, you can be sneaky, right? You can be a provirus and you can insert yourself into the host DNA. So we don't refer to these kinds of viruses as temperate viruses because there is no lytic cycle, right? They're only using the insert your DNA part, right? Only using the lysogenic cycle. They're not using it in combination with the lytic cycle, which is what temperate phages and temperate viruses typically do, is they alternate between the two. In this case, they're only using the one cycle where they are going to insert themselves into their host. There is no lytic cycle because, again, they don't lyse the cell when they leave anyway. But they can insert themselves into the host DNA. So now every time the host copies itself, it's copying the virus, right? Again, every time the host cell reads the DNA, it's going to transcribe that into RNA, which means now I have a strand that can be used as genetic material for the next infectious virus, right, that's going to leave the host. And I have a strand that can be used as messenger RNA to make, again, those capsid proteins and the surface recognition proteins. So I really have the best of both worlds if I am a retrovirus that functions as a provirus. And we have a really good example of this in HIV, which is, I have a little short video here coming up next that sort of describes the whole process of how HIV infects a cell and how it copies itself.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the two cell membrane, the cell membrane and the viral membrane together. And what ultimately happens is fusion of those two membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes in the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Okay, so to summarize how viruses replicate, and of course, again, remembering they can't do that themselves, right? They have to have a cell. They're obligate intracellular parasites. So viruses can get into cells either by injecting their genetic material directly, prompting those cells to undergo what's known as receptor-mediated endocytosis, essentially fooling the cells into thinking that there's something good, there's something nice that's knocking at the door, and then they wrap it in a membrane, bring it inside, and oh, lo and behold, it's a virus. Yay! Or, of course, by viral envelope. Phages can either use the lytic cycle if they're a virulent phage, or the lysogenic cycle if they're a temperate phage. We refer to viruses that infect eukaryotes just as viruses. So phages get the special name for working with bacteria. Eukaryote viruses, again, can use just the lytic cycle, so basically be a virulent virus like the rhinovirus that just uses the lytic cycle. Rhinovirus, of course, being the virus that causes the common cold kind of symptoms. And so the symptoms, of course, are your body's responding to cellular damage. They can use the lytic cycle and the lysogenic cycle, so something like the herpes virus that we were talking about earlier, or something like chickenpox. They can have the cell manufacture viral envelopes to leave the cell as an infectious particle, such as the case with HIV, for example. It's very common that these kinds of viruses are RNA viruses. Some may be retroviruses as well. Having them be DNA viruses that use a viral envelope is less common. And of course, proviruses. Again, these are viruses that insert their genetic material into the DNA of their host. Proviruses can infect eukaryote cells and stay in the genome even if they switch to the lytic cycle, right? So if they, which is unlike phages. Phages, when they're going from the lysogenic cycle to the lytic cycle, will actually remove themselves entirely from the host DNA when they're switching. Eukaryote proviruses don't do that. So where do new viruses come from, right? They sometimes seem to appear essentially out of nowhere, much in the way we saw COVID emerge onto the world stage in 2019, again, seemingly kind of out of nowhere. For the most part, they seem to come from mutations in existing viruses that may be held in reservoirs in different animal species, for example, and then they cross over to another species sometimes. We think that was the case with some of the viruses here on this particular list. For example, Zika, I think, and HIV. Notice HIV, it says 1959. That's the first confirmed case. They didn't realize it was HIV at the time until about a couple of decades later, when it really kind of exploded onto the world stage in the early 80s, the late 70s, early 80s. But looking back, folks had samples and discovered that, oh, actually, we had our first case way back in 1959 and didn't realize it at the time. Ebola, again, 1976, first case, and it erupts periodically. It has basically cycles that it goes through where it essentially seems like it's dormant in other populations, and then we get an outbreak in humans. So essentially, an outbreak that's localized is known as an epidemic. When you cross multiple continents, like we did with COVID, then you're dealing with a pandemic. So, for example, the influenza pandemic of 1917, 1918, where we had cases that began, I believe, in Europe and then again moved to North America and then spread elsewhere. If you're involving multiple continents, it's a pandemic. If it's fairly localized, then that's an outbreak. That's an epidemic. So like I mentioned, we think a lot of new viruses are coming from mutations in existing viruses. As we've seen with COVID, again, that thing seems to mutate like nobody's business. And it's an RNA virus. So RNA viruses are, as we saw earlier with when we were talking about replication, they often have to bring their own viral enzymes to accomplish replication of their genetic material, because their host cells don't have the enzymes to be able to accomplish that. So they have to bring their own tools to the party, and their tools don't have the proofreading capabilities that DNA polymerases, for example, have in living cells, which, as we're going to talk about later in DNA replication, we'll go back and check for errors. So generally, RNA viruses mutate at a higher rate compared to DNA viruses. Also, every time they replicate themselves, there's always the chance they're going to make a mistake. Well, when you have a very short generation time, right, meaning maybe only a few hours between infection and the emergence of the new viruses from the host cell, you can build up those mutations very, very quickly in a very short period of time. And so that, again, that helps to explain where some of these new viruses come from. And of course, like I mentioned, some viruses are crossing over from other species. So for example, the bird flu, H1N1, the bird flu, this is something that is present in some bird populations, and then it essentially jumped from birds to people, for example. So the animal populations can act as a reservoir. So this zoonotic behavior is not unique to viruses. We see it in parasites. We see it in some other organisms where they will jump from host to host. But it's not super common, but it does happen. So it's a very possible way of getting new viral infections. So how do we treat viral infections, right? So just like antibiotics are specifically formulated to attack bacterial cell processes, right? They interfere with cell wall formation, and they mess with bacterial ribosomes and bacterial enzymes in ways that they affect prokaryotes, right, bacteria, and they don't affect eukaryotes, right? So for example, if you have a fungal infection, an antibiotic is going to do precisely nothing towards that, because it's not going to affect fungi or eukaryotes, not going to affect those cells, right? So antiviral drugs then, of course, are going to have to be something that is going to interfere with virus replication inside the host cell without harming the host cell, which in most cases is us, right? And we're eukaryotes, or our fellow eukaryotes, right? And so we have two main classes of antiviral drugs. One are what are referred to as nucleoside mimics or nucleoside analogs. And these are molecules that are very, very similar in structure to normal nucleosides, right? So remember, nucleoside is the sugar plus the nitrogenous base, right? If you add the phosphate in, then it's a nucleotide, right? And then, of course, those are the building blocks for nucleic acids. So nucleoside analogs can be used by viral enzymes in replicating, for example, RNA, and they will not be used by host DNA polymerases. And essentially what happens is the RNA polymerase grabs a nucleoside mimic, adds it on to the chain, right, of the RNA that it's constructing, and then it goes to grab another nucleoside and discovers, oh wait, I can't form the bond, right? Because the nucleoside mimic is, again, similar in structure to a nucleoside, but it's not the same, and so it's not able to form that phosphodiester bond with other nucleotides. And so that essentially short circuits the viral RNA replication mechanism inside the host cell. They can't keep adding bases on because they won't bond properly. And then the other main sort of prong of attack for antiviral drugs are enzyme inhibitors. And you guys are going to get to play around with all kinds of enzyme inhibitors in lab. We're going to talk about them after we finish getting through metabolism, and talk about exactly how enzymes speed up and slow down reactions, or how you can slow down reactions by inhibiting enzymes. So in particular, enzyme inhibitors are going to target viral enzymes, right? They're going to target the viral RNA polymerases and bind to them in a way that causes them to not function the way that they're supposed to, essentially turning them off, or interfering again with reverse transcriptase or any other viral enzymes that are coming in the capsid. We can craft drugs that will, again, inhibit their function. And again, if the virus that's using those enzymes, if the enzymes don't function, then replication is not going to happen. Okay, so how do we categorize viruses, right? We have living things, we have, genus and species and all those levels of biological organization, but viruses are not alive, right? So we essentially categorize them predominantly based on the type of genetic material that they use, DNA or RNA, and whether it's single-stranded or double-stranded. So first of all, do not memorize this table. This is mostly here just to give you an idea of the range of different kinds of viruses that are out there in terms of what kind of genetic material that they're using. We also can categorize viruses based on how likely they are to cause disease or death, right? Their level of virulence. And of course, we can also categorize them based on how common they are, how likely you are to be exposed to them. So again, some viruses are very, very rare. Your chance of being exposed is very, very low. And then some, of course, are much more common. So again, just kind of giving you an idea. You have a range, double-stranded DNA, the top category here. We have several viruses that are, fairly deadly. There's pox virus, right? Smallpox, cowpox. These are fairly deadly viruses. Smallpox has actually been eliminated. Then you have others that cause chronic infection because they're proviruses. So the herpes virus, for example, we have several variations in that particular class of virus, right? Herpes simplex 1 and 2, varicella zoster, which causes chickenpox and shingles, and then the Epstein-Barr virus, right, which causes mononucleosis. And then, of course, you have some of the rather more severe papillomavirus, right, which can cause cancer, some other tumor-causing viruses. Another class that has a real big range of different kinds of viruses that are, some are, fairly benign and the others are really nasty, is the single-stranded RNA virus category, right? We have rhinovirus, that's common cold. Unless you are immune compromised, not likely to kill you. However, in the same category, we have poliovirus, which causes severe disease and sometimes death. We have the coronavirus categories, which include, again, the SARS and, of course, now, COVID-19, yellow fever, West Nile, and then rubella virus, which is a little bit more mild. It does cause birth defects, though, which is why we vaccinate against it. So again, in the single-stranded RNA category, category 5, we have things ranging from, actually, they're all pretty nasty in that particular category. There's rabies. There is no cure for rabies. I'll talk more about that in just a second. Ebola, which again is generally very, very deadly. We have had the first person survive Ebola recently. A few people survived it, and that was a major breakthrough. And so we're mining those folks for their antibodies. And then, of course, measles and mumps, which are other things we vaccinate against because they don't usually cause death, but they do cause severe brain damage in the case of measles. Which brings me to, what is a vaccine? Now, we are going to get into how a vaccine works later on in the course when we talk about the immune system. So I'm just bringing it up briefly right now so you sort of have an idea of what we're talking about. A vaccine is something that is using a weakened variant. So typically, for example, with viruses, they will disable it so that it is not able to infect cells by modifying its structure in a way that, again, it can't infect new cells. Or a derivative of a pathogen that's, so maybe, for example, just the spike protein on the COVID virus, for example, the essentially what's on the outside of the envelope that it's using to infect the cells. Or for case of bacterial vaccines, we often use basically we kill the bacteria and we use the dead bacteria as part of the vaccine. Basically, you want to present a weakened or inactive version of the pathogen to your immune system to stimulate it to mount a defense. So basically, you're going to show it something saying, hey, this is not you, and the next time you see this thing, you need to kill it with fire. And that's essentially what a vaccine is. It's prepping your immune system so that if it does encounter this pathogen, whatever it is, whether it's a bacteria or a virus, it can then respond overwhelmingly in a very short period of time to short circuit the infection and prevent you from getting sick. So it's essentially showing your body the infectious pathogen without actually infecting you. So you're not getting infected, you're not having to deal with virus parts, DNA lingering in your cells or damage or any of that kind of stuff. You're just stimulating your immune system to respond, which of course is why after you get a vaccine, right, you might have soreness in that area, you might run a little bit of a fever. That's your immune system responding to sort of this foreign particle. For most viral infections, we have treatments. They sort of work in varying degrees. That's one thing, of course, we discovered with COVID is some of the treatments that we had didn't work very well. And some of them worked better than others. And of course, thankfully, we've come up with new ones. We've discovered new ones that are more effective. And so we're better equipped to treat it now than we were, 2 years ago. But most viral infections do not have a cure. So we have to preventing a viral infection is really the key to keeping them under control. So how do we prioritize which viruses are worth investing in a vaccine for, right? And so this again brings back that idea of how common is the virus, how likely are you to encounter it in your everyday life, and how likely is it to actually kill you, right? So the common cold, obviously the word common, very common, but very much not likely to be fatal, right? And then you have things that are very likely to kill you, but are relatively uncommon. Your chance of exposure is low, for example, to Ebola, unless you are in certain parts of the world. Now, unfortunately, it's likely to be fatal if you are exposed and don't get a treatment, if that treatment even exists. So again, we've developed a lot of really good treatments for Ebola very, very recently. And then, of course, we have those survivors that we can then harvest their antibodies and use those to develop better treatments as well. Hantavirus, this is another one that, again, is your chance of exposure is pretty low unless you are in a place where you're sort of in the habit of coming into contact with the fecal material of deer mice or different kinds of mice that carry the hantavirus. I actually probably have antibodies to hantavirus from my field work, a couple of decades ago, because I was sleeping in a place that was frequented by deer mice, and of course we cleaned, but you never know what you at the beginning of the field season. Basically, people would come in with basically hazmat gear and clean it. But I imagine I've been exposed probably multiple times to that in very low levels. So I may very well have antibodies to that. And then, of course, you have the viruses that are both common and likely to cause lasting damage, if not death. And these ones, of course, are the big priorities for developing vaccines, right? So measles, pertussis, this is whooping cough. We had several outbreaks of that. I want to say a few years ago. It feels like 2016, 2017 was not that long ago, but we had big outbreaks of those. I remember they were pushing for folks to get vaccinated again because they had fallen off and weren't getting their kids vaccinated. And guess what? They were starting to get these childhood illnesses that, again, a child's body has trouble fighting, more trouble fighting off something like this than an adult usually does. And so you may end up with more damage. Again, measles can cause death, very definitely, and more often causes severe brain damage. So that's definitely something we want to avoid. And then, of course, rabies. This is something that is out there in a lot of wildlife populations, and you again, all of your pets should be vaccinated against it. If you're exposed and you're not vaccinated, you need to be treated within 24 to 48 hours. If you miss that window and the virus starts to replicate and move basically towards your brain, then it's 100% fatal. If it gets past that window. Your treatments are basically just supportive care, and I won't go into details, but it is a very nasty way to go. So definitely something you want to avoid. Again, vaccinate is important. And of course, we have lots of data to demonstrate that vaccines work, right? Here I've got a report of, again, the whooping cough outbreak that we had, again, relatively recently, 2013 or so. But this is tracking whooping cough cases back to 1920. And as you can see, fairly low, 1920, 1930 in Texas. Then you have, the 40s, you get the baby boom, right? More people, more crowded conditions, and so great conditions for transmitting a virus around. You notice the whooping cough vaccine developed right around 1949, 1950. And oh, look, things start falling off and down to close to zero. Again, not everybody can be vaccinated. And then, of course, folks said, oh, well, you know what? Nobody gets whooping cough anymore. Nobody gets measles anymore. And unfortunately, the reason for that is that most of the populations, upwards in some cases 90%, had been vaccinated. And that's why they weren't getting it, right? They were definitely exposed. It's still out there in the natural world, but your body fights it off before you really know that you've been exposed, right? And the only virus that I know of that has been eradicated, again, they talk about, oh, we eradicated measles. Well, here, but then it's coming over with, folks coming over from other countries, so it's not eradicated globally. The only one that has been eradicated globally is smallpox. Now, it still exists in 2 classified facilities. I want to say at least 2 countries, probably 3. We have some, again, in a classified military medical lab. Russia has some, and I believe there is some in West Germany. I could be wrong about that last bit. And the reason that's scary, well, it's scary and it's not, is we also have vaccines. The vaccine, whatever the vaccine is made from, my brain is blanking on the word. We have those as well. So we could crank out the vaccine, but as we've seen with COVID, that's a process that can take a little bit of time. And so we should get up and running. The scary part, right, is we don't vaccinate people against smallpox anymore. My husband's brother, who was born in 1970, has a smallpox scar. My husband and I do not, and he did not have a smallpox scar. We were not vaccinated against smallpox because, again, by then it had been eradicated. And they said, okay, you don't need it because your chance of exposure is basically 0. It's gone. So if someone decides to steal this and weaponize it, we could be in for a world of hurt. So we just hope that that doesn't happen. And on that happy note, we are done with this EdPuzzle.